folks. Hello and welcome. This is the Apologetics Academy, a weekly webinar which I run every Saturday, normally at 8 p.m. here in the UK, which corresponds to 3 p.m. Eastern, 2 p.m. Central, or 12 noon Pacific. Each week we bring on different scholars, apologists, experts, thinkers, intellectuals from across the theological and philosophical spectrum to present on topics of interest to Christians, particularly pertaining to the question of whether or not Christianity is true. And we also cross-examine and evaluate competing worldviews such as atheism, Islam, Mormonism, and so on. There will be, every week we give opportunity for our special guest to give a presentation, and then we open it up to open floor Q&A, discussion, dialogue, and debate and interaction. There's a number of ways that you can participate in today's webinar. You can simply watch and listen anonymously if you so wish. There's also, you may have noticed, a chat window where you can type in chat as the webinar is in progress. You can also submit questions in textual form by hitting the Q&A button at the top of your screen and typing in a question. You can do so anonymously if you wish. You can also click the raise hand button, and I will promote you at the appropriate time to be a co-panelist with our guest speaker, and you'll have the opportunity to interact and engage and discuss. And people can take it in turns to have a conversation with our speaker. Just to give you an idea of what's coming up in the coming weeks, next Saturday on March 11th, we have a very prolific astrophysicist by the name of Dr. Luke Barnes with us. Dr. Barnes is an Australian physicist. He is probably the world's leading expert on the fine-tuning argument for God's existence, and he's going to be talking to us about the thesis of his latest book, A Fortunate Universe, Life in a Finely Tuned Cosmos, making the case for God from the fine-tuning of the initial parameters and constants of our universe. The following week, on March 18th, we have with us Dr. Richard Weikart, who is a famous historian, who's going to be talking to us about his book, The Death of Humanity and the Case for Life, which argues that secular philosophies have eroded the Judeo-Christian sanctity of life ethic. And then the following week, on Saturday, March 25th, we have with us Dr. Paul Nelson of the Discovery Institute, who's going to be talking to us about evolutionary theory. Dr. Nelson's a philosopher of biology, and he's going to be talking to us with the subject title, Evolution is a Theory of Transformation, Not Similarity. And then finally, the following week on April 1st, we have a very famous atheist philosopher speaking to us called Dr. Stephen Law from the University of London, who will be making the case for atheism. So back to today's session, we have with us Dr. Scott Minnich, who is a prestigious bacterial geneticist from the University of Idaho, who is an expert on Yersinia. You might recognize Yersinia pestis as the agent of bubonic plague. And Dr. Minnich is an expert on the type 3 secretion system in particular, and also the flagellum. And he is also interested in long-term evolution experiments, particularly those conducted by Dr. Richard Lenski, who is a microbiologist at Michigan State University. And this evening, he's going to be talking to us about a paper which he published, basically reviewing that work and reviewing its implications for the origins discussion, debate. So Dr. Minnich, do you want to maybe just introduce yourself, just tell us a little bit about your background, and then just launch into your presentation? That would be awesome.
Okay, thanks, Jonathan. It's good to be here. And so, in your introduction, yeah, I think microbial genetics has been my training. I got my PhD at Iowa State University and postdoc at Purdue and Princeton, and then came to the University of Idaho in 1989, where I worked primarily on Yersinia and host parasite interactions. I have been interested, off and on in Lenski's long-term evolutionary experiments, and I think their publication in 2008 caught my attention when they had published that E. coli had gained a novel, innovative gain-of-function mutation and allowing it to grow on citrate. So, you know, and looking at that and reading about it, it dawned on me that there might be another explanation. And so I think these experiments were prompted by listening to him in Boston in 2014 at American Society for Microbiology meetings, and decided to just, you know, test our hypothesis that there was a simpler explanation to the citrate mutant that he isolated. So I thought what I'd do today is just go through a 30 to 40 minute PowerPoint presentation, kind of outline the problem and our response to it and our interpretation, and then we can open it up for questions. How does that sound?
That sounds awesome.
Okay, so I want to share my screen here, see if I can do this correctly. And then I want to make sure I share the computer sound. Okay. All right, can everybody see this?
Yep. So then start the presentation and it should appear full screen.
Okay, so all right, here we go.
There you go.
All right, so the title of this talk is Reinterpreting Long-Term Evolution Experiments, Historical Contingency versus Selection. And I want to acknowledge collaborators on this. Actually, most of this work was done by Dustin van Hofwegen, who's now at the Rocky Mountain Labs, NIH Labs in Montana, as a postdoc, and then collaborator Carolyn Hovde-Bohach, a distinguished professor here at the University of Idaho. Okay, so outline of the talk, I want to introduce Lenski's interpretation of mutation to the CYP+ phenotype. Aerobic citrate utilization is a rare innovative gain of function event, as they claim. They also attest that CYP+ requires a series of neutral preceding mutations, this idea of historical contingency. It touches on a long-term question in evolution that goes back to Darwin, whether the strength of selective pressure can affect the frequency and the specificity of adaptive mutations. So our counter hypothesis we'll present is that CYP+ mutants can be rapidly and repeatedly isolated. Historical contingency is not a requirement, and the strength of selection does not affect the frequency nor the specificity of adaptive mutations in this case. Now, for those of you that aren't familiar with these experiments, I want to show a short video, a 4-minute video that was aired, I think, about 2 years ago, and let Lenski explain his own work and narrated by Morgan Freeman on Through the Wormhole. So hopefully this will work.
Richard doesn't have billions of years to watch evolution in action, but he does have the span of one lifetime, which he has dedicated to performing the longest evolutionary experiment in the history of science. 27 years ago, Richard plucked 12 genetically identical E. coli bacteria from a container and allowed them to start their own populations in 12 separate worlds. Ever since, day after day, he's watched each of the 12 populations multiply and compete amongst themselves for a limited amount of food.
Key part of the experiment is just keeping it going day after day after day. transferring a small amount from each flask into fresh medium each day. And the bacteria grow and replicate in that fresh medium until they run out of the sugar that they grow on. And then they sit there until the next day that whole process is repeated.
At the end of every day, he allowed a small sample of the survivors to continue producing new generations. Year after year, the daily process went on and on. In each flask, the cells of bacteria replicate, passing on tiny random mutations to their offspring's DNA. For the first 15 years of the experiment, the separated populations evolved pretty much in lockstep, each strain gradually adapting to this flask environment. But one night, one of the colonies had a sudden and dramatic victory in the arms race of evolution.
One day we came in and found that one of our flasks had many more bacteria than the others. One of the lines of bacteria, after they'd finished the glucose, they said, there's something else to eat. And that gave them a huge advantage.
Overnight, these E. coli had switched on new and complex internal machinery that allowed them to extract energy from a benign chemical additive. Armed with this new talent, they vanquished their competitors. But when Richard examined the DNA of these E. coli, he realized that the mutation that switched on the new talent was merely the last in a long series. The others had lain dormant for thousands of generations. Richard thinks of it as life playing a grand version of his favorite game, poker.
I always liked playing games that involved a mixture of luck and skill. Most card games, you've got the shuffling, and yet poker takes some skill to make a lot of money.
For E. coli and humans alike, each genetic mutation is like a new card dealt to the species. If the right hand is made in the right order, the organism can win big for posterity.
In this case, the bacteria have this key mutation, but it had to happen in the context where there were other mutations before it.
When there is a queen and a king of hearts on the table and you have the 10 and ace of hearts in your hand, you are set up to potentially make a royal flush, the most powerful hand in poker. All you need is for the final card to be a jack of hearts.
Well, if you were looking for a third 7, that does you no good at all. But if you got the other parts of a royal flush, you're happy.
E. coli adapt to meet their immediate needs through genetic mutation. But some mutations don't have an immediate reward. They are investments for their distant descendants, so that they can win a big evolutionary jackpot.
Okay, so that's the experiment that's been ongoing since 1988. And just to review what Lenski has done, and these are excellent experiments. He took isogenic culture of E. coli, split it into 12 flasks, and on the left here, he's using a minimal medium with glucose as the sole carbon source at a concentration of 0.0025%. So this is a low concentration of glucose. Usually we use 0.2% when growing organisms. And, whoa, I'm getting ahead of myself here. This allows 6 to 7 generations, and every day for the last 28 years, they've transferred all 12 of these cultures and monitored their growth. Every 500 generations, I don't know why this came up. Okay. Every 500 generations, you can take a sample out and freeze it. So you have revivable frozen fossils. You can look at fitness costs by doing competitions between ancestors and their siblings. With the advent of genomic sequencing, you can mark mutations to the nucleotide level. So you have these rapid generation times and high population numbers. Okay, so it's an open-ended experiment, and as of July of last year, they had approached 65,000 generations of E. coli in this experiment. So this is a Herculean effort, and it's still going on. In an article in Science in 2013, Lenski's work was highlighted. At this point, they had gone through 58,000 generations, screened 10 to the 14th bacteria. So that's 100 trillion cells, 25 years, $4 million in costs, 75 person years, 30 participating graduate students and postdocs, and over 10,000 liters of media. In a human equivalent, this is really interesting. This is with a generation time of 15 to 20 years. For humans, it would be about the equivalent of 1.3 million years of human evolution, 10 to the 11th individuals, which is 10 to 100 times the estimate of the number of people that have ever existed on this planet. So these are an excellent experiment to look at the adaptive capability of an organism over an extended time period, out to 65,000 generations. Okay, the key observation occurred at 15 years, or approximately 33,000 generations. If you recall from the video, Morgan Freeman remarked that all 12 cultures were essentially in lockstep. In fact, Lenski had published that perhaps they had disproved Stephen Jay Gould's theory of contingency because they were acquiring mutations in the same gene, going down the same adaptive pathway, until this key point in the evolution of these cultures. And one of the 12 suddenly bloomed and reached higher cell densities, which you can see here in population A3 when you look at the turbidity of this flask compared to the others. Something had happened. It had learned to grow on this benign substrate that Morgan Freeman mentioned in his video. That's citrate. Okay, so as this was reviewed in the press, in the New Scientist, Bob Holmes wrote, "A major evolutionary innovation has unfurled right in front of researchers' eyes. It is the first time evolution has been caught in the act of making such a rare and complex new trait." Zachary Blount and Richard Lenski have even intimated that this could be perhaps a true speciation event, because citrate utilization for E. coli is a phenotype that we use diagnostically to differentiate it from closely related coliform organisms. Lenski himself is quoted as saying, "The most dramatic change we have observed to date is the origin of the new ability to grow on a citrate." In the video, it also mentioned that this phenotype, this final step in gaining access to citrate, was just the last step in a series of mutations that led to this outcome. And this is where the idea of historical contingency comes in, that there are preceding neutral mutations that don't show a phenotype but are necessary for the outcome of citrate utilization. So this quote, "If evolution is highly contingent in the unpredictability sense, then one might expect evolutionary outcomes to be fundamentally unrepeatable, even from the same starting point." Zachary Blount last year wrote in this paper, "A Case Study in Evolutionary Contingency." Perhaps the most intriguing and important question about the evolution of CIT+ is also the most obvious. The enormous citrate resource had been there from the LTE's beginning. So why did the CIT+ trait evolve only once, and then only after such a long time? One plausible explanation is that the CIT+ was a historically contingent trait. Historically contingent traits require particular non-guaranteed antecedent states, which is to say a particular history, to evolve. Their origins are therefore complex and require multiple mutational steps. I took this figure, or Dusty did, this is part of his presentation, out of a paper by Quant in Barrick's lab in Texas, just showing the evolution of this CIT+ trait and how it's been explained by the Lenski lab and others. And there are 3 stages. The first stage is referred to as potentiation. This is the long period of acquisition of these neutral mutations that set this E. coli up for actualization. So if you look at the phenotype down here, in this potentiation stage, the cells are CIT-. They can't utilize citrate. But you reach this stage, and this was at 31,500 generations, where they became weakly CIT+. You get a little tick in the amount of the cell density in this one culture. And then a little over 2,000 generations following that, you get this major increase in cell density in this one of 12 populations of cells. And this is referred to as refinement. So again, at 31,000 generations, you're weakly CIT+, and then at 33,000 generations, it's refined, it's fixed in the population, it sweeps through and outcompetes the other parental cells. And this required a mutation in DCTA. This is a dicarboxylic acid transporter, which I'll talk about in a minute. Okay, so potentiation, actualization, weakly CIT+, and refinement. Now, when these were analyzed by genomic sequencing, this mutant was analyzed, what Lenski and Blount found was, here's the wild type genetic loci that's involved. You have CIT-G and CIT-T as part of an operon. CIT-T is a transporter for citrate. It's only expressed under anaerobic conditions. These cultures are all being grown aerobically, but the CIT+ mutant was a promoter capture. So you had amplification of this region in the genome and a recombination event that now you have this duplication of CIT-T, and this gene is now under the promoter of RNK. So that's driving this gene, allowing it oxic expression under aerobic conditions. All right, so we refer to these as promoter capture. All right, so that's what happened in their estimation. You have this promoter capture at 31,000, and then you require a second mutation in this dicarboxylic acid transporter, DCTA. But these were all set up by these potentiating mutants, which has been the subject of study over the ensuing years. What are these historically contingent mutations that are required to allow this transition to CIT+? Okay, so questions that I wanted to consider and doubted was, are historically contingent neutral mutations necessary? And I'll give you a hint of why we were thinking that. And also, does the strength of selection affect the specificity and the frequency of adaptive mutations? Because they have alluded to this as well in some of their publications. Now, why would I consider or doubt historical contingency being required? It's because in 1982, Barry Hall, another microbial evolutionist, was trying to get E. coli to grow on the substrate phenylarabinose, which E. coli can't grow. He was trying to isolate mutants that extended the substrate specificity for a beta-galactosidase. So he put E. coli K12 into this, essentially the same media as Lenski, with phenylarabinose is the sole carbon source, allowed them to grow aerobically. After 14 days, this culture bloomed as well. But as Barry Hall found, they weren't growing on the phenylarabinose as intended. They were growing on the citrate that's added into these media traditionally as the chelation agent. It can bind iron and transport that to the cell. So here's a case where Barry Hall isolated the same phenotype of E. coli in 14 days, where it took Lenski 15 years to get the same isolate, and only once in 12 parallel cultures. Now let's look at citrate metabolism quickly to bring some of you to speed if you're not familiar with metabolism. If you feed E. coli glucose, it will break it down through the Embden-Meyerhof pathway to pyruvate, which is then converted to a 3-carbon compound. So you start with a 6-carbon compound, you get 2 pyruvates, you split off a CO2, and the other 2 carbons are converted to acetyl-CoA, which condenses with oxaloacetate to form citrate, and you go through the TCA cycle. So essentially, glucose under aerobic conditions can be oxidized completely to water and CO2 through these steps. Electrons are collected by electron transfer molecules fed into the electron transport system to generate ATP. Now, E. coli can make citrate. It can metabolize citrate, but it can't grow on citrate aerobically because it can't get this carbon compound into the cell. So over here, we have under aerobic conditions, there's a block, but under anaerobic conditions, E. coli can grow on citrate because it expresses CYP, which is an anti-porter. It will import citrate into the cell, but it requires an export of this 4-carbon dicarboxylic acid, succinate or oxaloacetate. When E. coli under anaerobic conditions imports citrate, it's hydrolyzed to acetate and oxaloacetate. Acetate can be used as a carbon substrate. E. coli can grow on acetate, but there are only 2 carbons. And because CYP is an anti-porter, you have to have succinate to export to drive citrate import. So all the electron or reducing power generated from acetyl-CoA, is used to reduce oxaloacetate back to succinate. In essence, it's a wash energetically, and that's why to grow anaerobically on citrate requires another co-metabolite to provide reducing power. And this was all worked out in the 80s by Lutkins and Gottschalk. I mention that because Lenski criticized us in saying there was new information involved in our mutants because E. coli requires a co-metabolite normally, but it doesn't in his mutants and our mutants that we subsequently isolated. But that's just really a reflection of the conditions, the environmental conditions, and the difference between fermentation and respiration. We can come back to that later. But in looking at Hall's experiment and Lenski's and the difference, I could visualize this as a difference between a direct selection, where E. coli only has citrate as a potential carbon source in the environment, versus Lenski's experiment, which we term a genetic screen. So direct selection are conditions in which only the desired mutant can grow, whereas in a genetic screen, so he's got glucose plus citrate in the environment, both glucose utilizing and potentially citrate utilizing organisms can grow, but they're going to be in competition. I wrote Lenski in 2012 when they identified this duplication in CYP as responsible, this promoter capture, thinking that there may be another explanation. You might get the same mutants in a direct selection. But he published a paper in 2013 what I find interesting. If selection is too strong, okay, so this would be equivalent to a direct selection, then an evolving population might be driven towards a quick fix, a local peak in fitness landscape that renders an even better solution less accessible. In other words, he's saying the difference in selective pressure can affect perhaps the speed, but in this case, the type of mutation that's acquired. A strong selection is going to give you just a quick fix, whereas a long-term experiment will give you a better solution. This is testable. And one of the reasons why I wanted to do the experiment. So our hypothesis was that CIT plus E. coli, citrate utilizing E. coli, can be rapidly and repeatedly isolated. This we could rule out historical contingency as an involvement, all these antecedent mutations being required. And will follow the same genetic trajectory identified by the LTE, showing selection strength yields the same class of mutations. So we set out to do it. We did first an experiment to see if we could repeat Barry Hall's experiments with E. coli K12. So here's a graph. We're looking at the number of organisms per mil in a 50 ml flask, where citrate is the only substrate available to these organisms over time. And we found, and we used wild type E. coli K12 in this experiment, because these organisms are essentially in a stressed starvation condition equivalent to stationary phase growth. We included a mutant as a negative control, mutation in RPOS. This encodes the sigma factor required for growth. In stationary phase, it activates DNB, an error-prone polymerase that can perhaps accelerate the acquisition of mutations. So we included that to see what would happen. And also 2 mutations, one with a deletion for the CIT transporter. Is there another solution to this problem? And a deletion in DCTA, the succinate importer, to get succinate back into the cell. So in other words, CIT will function, bring citrate in, but you lose 4 carbons. For the 6 that you bring in, you lose 4. You have to recapture succinate to grow efficiently. And we found that after, and we repeated this a number of times, so we got a royal flush in all 3 of our cultures with our wild type within 21 days. Surprisingly, we found that our RPOS mutant dropped almost 3 logs, 1,000-fold by day 28, but then began to recover. And we had 3 of, I think, 4 cultures that did that, but we had no CIT plus mutants in our CIT deletion mutant, showing that CIT is required, nor in DCTA, although we found 1 of a number of strains after 139 days, actually in our DCTA mutant regained the ability to grow on citrate. So the bottom line is we recovered, like Barry Hall, in days versus decades of Lenski's experiments, CIT plus E. coli. Now the question is, are they really using citrate? And we did this by mass spectrometry. And here we have a graph on the left side. We're measuring citrate concentration at micrograms per mil. So we're starting out a little over 2 micrograms per mil. And on this side of the graph, we're monitoring optical density, our growth of E. coli, and our scale down here is in hours. So once we got a CIT plus mutant that was refined, efficient, we began this experiment. And you can see that as E. coli starts to grow, the concentration of citrate drops. As we reach exponential phase, then you have exponential utilization of citrate. It's completely used up, and E. coli stations out in stationary phase. All right, so this was a refined CIT plus mutant. In our paper, it was DV133. And to look at contingency, another way to look at contingency, because this mutant took a number of days, are there other mutations on the chromosome required for this phenotype? We took a transducing phage, bacterial virus, grew it on this mutant, and then transferred the CIT plus phenotype into the wild type E. coli background. So this will have, it's never been exposed to citrate for that length of time. It doesn't have those antecedent mutations. And we find that that mutation was faithfully transferred, and these organisms grow well on citrate as well. So the fact that we can get these mutations in as few as 12 generations to 40 generations, and the fact that we can transduce that mutation back into the wild type genetic background, in my mind, rules out contingency. All right. When we did genomic sequencing on this first mutant, we found that the CIT transporter had acquired an insertion element right upstream to provide an outside promoter to drive CYP aerobically. And this was the mutation that was transferred into the wild type background. The predecessor for this refined mutant, we also sequenced, and it just had large duplications of this region of the chromosome of over 100 kb and up to 8 copies of CYP. So you had gene amplification of this region, and then that was refined by a second promoter remodeling to capture CYP behind this insertion element. It's interesting, in replay experiments that Lenski has done with Zachary Blount, they have acquired this same class of mutations in their replay experiments. Another one of the mutants that we isolated that had refined had a duplication of CYP. You recall Lenski's initial mutant had CYP now spliced behind the RNK promoter here in gray. In our mutant, very strong CYP positive phenotype, had simply recombined CYP behind the USP gene here instead of RNK, so a little bit further downstream. USP is one of the most highly expressed genes under stationary phase of growth. So again, just to remind you, this is Lenski's single mutant out of 12 parallel cultures, and they had a duplication that positioned CYP behind the RNK gene. The bottom line is that we isolated these mutants rapidly, and we're getting the same class of mutations that Lenski got in his long-term experiments. And we can get these repeatedly, okay, from our direct selection. Now, Dusty had this idea that let's go back and repeat Lenski's experiments, the same conditions, this media, low concentration of glucose. So our previous experiments that I just described only had citrate in the media. Let's repeat Lenski's experiments, but because gene duplications are involved and they're inherently unstable, we hypothesized or speculated that Lenski was not isolating these mutants because with an everyday transfer to fresh glucose media, even though these duplications could be occurring as a background to subpopulation of cells, once you put them into fresh glucose, now it comes with a fitness cost, and they'd either be recombined out because it's a reversible mutation or outcompeted because of the cost of carrying extra DNA. So we repeated these experiments, but instead of transferring every day, we transferred once a week, and we monitored these cultures for growth. All right, so this is essentially the same experiment of Lenski's, same conditions, but we're just allowing the organisms an extended period in stationary phase to see if we could lock in these gene duplications. We do the experiment. We call this a modified direct selection in our paper with glucose, and we found that this extended time between transfers, 7 days versus 1, yielded CYP+ mutants in less than 35 days. So here again, we're looking at optical density, turbidity of our culture, monitoring growth. We start out with a low concentration of E. coli, and on this axis of the graph, we have days. At every week, we are transferring these cultures into fresh media with glucose, and we found after the third transfer that we get robust recovery of CYP+ cells. We did this 9 out of 9 times for wild type. This also occurred, took a little longer with our mutant inhibited in stationary phase. We never isolated mutants with deletions for DCTA or the CYP transporter. Again, this is not required, this mutant or phenotype does not require historical contingency. We were dealt a royal flush every time we did the experiment. Here's a summary where we start out, we're looking at the number of cells and density. And we're transferring in the LTE every day, in our experiment every week, and after the second, third, or fourth transfer, we get CIT plus mutants easily. So a model to explain the recovery of CIT plus phenotypes in both our experiments and the Lenski experiment. It follows these points. First, prior to selection, growth in glucose, a subpopulation of E. coli carry duplications in the citrate region of the chromosome. Duplications are the most common mutations that occur. 1 to 2% of a population of E. coli is going to be duplicated for any region of the chromosome. This duplication is large, flanked by insertion sequences, which can foster further CIT amplifications, CIT-T amplifications of this region of the chromosome. Increased copies of CIT-T favor growth in citrate. I think the phenotype or these gene expression is leaky, but sufficient to allow some growth during the stationary phase. But these duplications or amplifications are constrained by their high segregation frequencies when transferred back into glucose medium. All right, they come with a fitness cost. They're unstable, they're reversible. But with extended incubation in stationary phase, cells favor CIT plus because there are more target CIT plus alleles, CIT-T alleles, are added to each developing clone. In other words, you have more cells and more copies of CIT-T per cell. This amplification state increases the probability of a second event, chromosome remodeling, to capture a nearby constitutive promoter. Once remodeling occurs, all CIT plus alleles are subject to loss by segregation, minimizing the instability of multiple gene copies and thereby fixing a stable CIT plus phenotype in the cell. Okay, so in summary, E. coli underwent rapid adaptation to aerobic citrate metabolism. Dusty isolated 46 independent CIT plus mutations in the short course of these experiments, compared to 1 mutant of CIT plus in 15 years in the long-term evolutionary experiments. Potentiation actualization of CIT plus phenotype occurred within 12 generations. Refinement required fewer than 100. The genetic trajectory of this adaptation and the classes of mutations identified centered on CIT-T and DCTA expression via gene amplification and chromosome remodeling, promoter capture of CIT-T. Okay. Importantly, we showed that there's no difference in the class of mutations between a strong direct selection and a weak long-term genetic screen. And to me, this is the most satisfying result of our experiments. We ended our paper saying this controversial statement, E. coli's capacity to evolve is more limited than currently assumed. And although the long-term evolution experiments give the impression that the transition to CIT plus is a rare, innovative gain-of-function mutation requiring a number of neutral mutations before this phenotype is achieved. We think that our experiments show the solution to the same problem can be rapidly achieved. In other words, there are very few solutions to this problem, maybe only one. We liken these experiments to the Greek mythological account of Sisyphus, if you remember, who was condemned to push the stone up the mountain for eternity, from Homer. And I saw Sisyphus at his endless task, raising his prodigious stone with both his hands. With hands and feet, he tried to roll it up to the top of the hill, but always, just before he could roll it over onto the other side, its weight could be too much, would be too much for him, and the pitiless stone would come thundering down again onto the plain. Then he would begin trying to push it uphill again. All right, so E. coli under the LTEE would have a short period in stationary phase, trying to push themselves over the edge, and then The stone would roll down the hill. They're transferred into fresh medium, and they've got to start all over again due to the fitness cost of gene duplications. So everyone in microbial genetics learns early on this famous quote by David Botstein: "A selection is worth 1,000 screens." This is illustrated showing Zachary Blount and the effort to isolate these mutants, this mountain of petri dishes and Dusty on the right-hand side here with a few flasks, a few tubes, a few plates, and we get to the same result easily. So with that, I'll open it up to questions. If you have any.
Sure. Thank you, Dr. Minnich. Do you want to unshare your screen there so that we can?
Yeah. How do I go back to?
Be at the top of your screen. Should be a button that says Stop Share.
Okay. Stop share. Okay.
Right.
Got it.
Awesome. Well, folks, this is the interactive portion of the program. If you would like to interact with Dr. Minnich, you can submit questions via the Q&A box at the top of your screen, or you can also click Raise Hand, and I'll promote you to be a co-panelist and engage and interact that way. We have a question submitted from an anonymous viewer who asks, how do the large numbers of microbes provide evolutionary opportunity contested with the limited opportunity of smaller populations like plants, reptiles, amphibians, mammals, etc.?
Okay, I repeat it one more time. I got some of it was blurred.
Sure. You can also read it yourself if you hit the Q&A button at the bottom of your screen. But it says, how do the large numbers of microbes provide evolutionary opportunity contested with the limited opportunity of smaller populations like plants, reptiles, amphibians, mammals, etc.?
Okay. Oh, I think, that's a good question. And the advantage which Lenski and others have taken with microbes is they have these rapid generation times, hours, and you can reach high cell densities, and you can monitor genetic changes, evolution over, easy span of time. So if that answers the question, I think that the experiments are just the ones to do. I mean, it's the perfect model system to see the limits or capabilities of any organism to adapt and change over time.
Cool. We had a hand raised from Bill Cole. Bill, you're live with Dr. Minnich. Welcome. Go ahead.
Thanks, Scott.
Yeah.
Pleasure to meet you.
Howdy.
A question I have is as far as I understand, I don't know much about bacteria, but E. coli has very strong DNA repair mechanisms. Is that right?
Correct.
And so my conclusion here would be that maybe what you're seeing is non-random. That it would naturally repair this, but that there are mechanisms inside the bacteria that are dealing with stress and creating these mutations on their own, due to their own mechanisms and not random change. Just
yeah, in a sense, although it is true that E. coli DNA replication has high fidelity. And in part, that's why we included the RPOS mutant, because, under stress, you induce an error-prone polymerase that can increase the mutation rate. And we wanted to see if that was involved in this process, and it doesn't appear to. But the bottom line is, to get these mutants, you simply have to amplify the gene required, this citrate transporter, and then recombine it. Once you have this region amplified, you can have this chromosome remodeling and get it fused behind an oxic promoter, and that occurs pretty rapidly. And this is I think what really prompted me to do these experiments, because it was very reminiscent of experiments that John Cairns did in the late 80s with E. coli, in which opened up this whole question of directed mutagenesis. Can cells sense their environment and direct mutations to solve, this problem of stress or whatever? And this conflicted with the early experiments of Lurie and Delbruck, which essentially said that all mutations are pre-existing in a population. And I think John Roth solved that problem. It wasn't directed mutagenesis. It was just the same mechanism that we're seeing here, amplification and then chromosome remodeling to fix an efficient mutant, whether it's growth on lactose or growth on citrate in this case.
But why wouldn't the repair mechanism, if it was random, take it out and bring it back to its normal configuration?
Well, once you have this duplication and it's fixed,
well, how does
you can have a reverse mutant.
How would the duplication get fixed given the repair mechanism?
Because there's selective advantage for these cells to grow in this environment, even though they're weakly CIT+. You're selecting for them, and that's the whole point. As these cells grow slowly,
it's very high pressure selection, in relative to most elements. Got it.
Right, and it's the same selective pressure under Lenski's experiments, but these duplications indeed come with a fitness cost. So once you put them back into glucose, these cells that have an amplification of CIT are at a disadvantage, just due to the amplified DNA, and you do get rid of them.
Yeah.
You recombine them out. It's just recombination.
That's a very important point from an evolutionary standpoint, that there's this reversion to the mean. Right.
It's a reversible phenotype.
Yeah.
It's not like antibiotic resistance or phage resistance. Once you get the mutation, you've lost that phenotype, but you're resistant to the phage.
Cool.
Okay, is that all from you, Bill? Okay, thank you, Bill. We have a question submitted from an anonymous viewer. An anonymous viewer asks, I teach honors bio, AP bio, and introductory college bio. How do I communicate ID is supported by a lack of historical contingency? Do I share that the mutant is already there in the genome? It might seem to the students the contingencies make the CIT+ more difficult to achieve, thus extending any evolutionary time factor. Thanks for clarifying.
Okay, I mean, that's a key point that Lenski has made, that the transition to citrate utilization is dependent upon a number of silent mutations preceding it. Okay, so this just merely, as Morgan Freeman said, merely the last step in a long series of mutations. And their experiments understandably give that impression, because they only got one of these mutants after 15 years in 12 parallel cultures. All right, our experiments show why that's not the case. It's a matter of experimental conditions. It's an artifact of the experimental conditions that we can get these mutants in 12 to 40 generations. There's no time to acquire these necessary antecedent mutations, and the fact that we can transduce the mutation into the wild type clean genetic background shows that these mutations, potentiating mutations, are not necessary.
Okay. Again, just a reminder, if you want to interact with Dr. Minnich, you can either submit questions via the Q&A button at the top of your screen by entering them in textual form, or you can also click the raise hand button to be made a co-panelist. While we're waiting for more questions, let's just perhaps run through what Rick Lenski said in response to this work, as you or where he made a blog post. Do you want me to read portions of the blog post and you can respond to it, Scott? Or
sure.
Yeah. Okay. So let's just run through it point by point. So he starts off, those who follow the long-term evolution experiment with E. coli know that the most dramatic change we have observed to date is the origin of the new ability to grow on citrate. It's dramatic for several reasons, including the fact, external to the long-term evolutionary experiment, that E. coli has been historically defined as a species based in part on its inability to grow on citrate in oxic environments, and the fact, internal to the long-term evolution experiments, that it was so difficult for the bacteria to evolve this ability that only one of the populations did so, and that it took over 30,000 generations, even though an abundance of citrate has been present in the medium throughout the long-term evolution experiments. Even after 64,000 generations, only the ARA3 population has evolved that new ability. Zachary Blount, formerly a graduate student and now a postdoc in my lab, has spent the last decade studying the evolution of this population and its new ability. His two first authored papers in PNAS 2008 and Nature 2012 demonstrated, respectively, that 1, the origin of the ability to grow on citrate in the long-term evolution experiments was contingent on one or more potentiating mutations that happened before the actualizing mutation that conferred the new function first appeared, and 2, the actualizing mutation was a physical rearrangement of the DNA that brought together a structural gene, CYP, that encodes a transporter and a previously unconnected regulatory region to generate a new module that caused the phenotypic transition to CYP+. These papers presented and discussed much more than these 2 points, of course, but they are the key findings. More recently, Zach was a co-author in a paper in eLife 2015 by Eric Equant, Jeff Barrick, and others that identified 2 mutations in the gene for citrate synthesis, one that potentiated the evolution of citrate utilization and another that subsequently refined that new function. So we were keenly interested when we saw a new paper titled Rapid Evolution of Citrate Utilization by Escherichia coli by Direct Selection Requires CYP and DCTA by Dustin van Hofwegen, Carolyn Hovde, and Scott Minnich. The paper is posted online in an accepted manuscript by the Journal of Bacteriology. What follows here are some overall impressions of their paper that Zach and I put together. We may follow these impressions later with some further analysis and comments. So this is how they respond. They said, let's begin by saying that it's great to see other groups working on interesting systems and problems like the evolution of citrate utilization in E. coli. Moreover, the actual science that was done and reported looks fine and interesting, though we have a few quibbles with some details that we will overlook for now. By and large, the work confirms many of the findings that were reported in our paper cited above. 1, the ability to grow on citrate in the presence of oxygen can and does evolve in E. coli, Blount et al. 2008. 2, when aerobic growth on citrate evolves, it does not do so quickly and easily, Blount et al. 2008, but instead it takes weeks or longer, more on that below. 3, all strains that have evolved this new ability have physical rearrangements that involve the CYP gene and appear also to involve a so-called promoter capture, whereby a copy of this transporter encoding gene acquires a new upstream regulatory region, Blount et al. 2012. And 4, genetic context matters. The strain one uses affects the likelihood of evolving the CYP+ function, Blount et al. 2008, and the resulting ability to grow on citrate, Blount et al. 2012, Quant et al. 2015. The problem, then, is not with experiments and data. Rather, the problem is that the results are wrapped in interpretations that are, in our view, flawed and fallacious. So then they put forward some of their reasons for thinking that the interpretation is flawed and fallacious. So maybe we can run through those. So the first one is no new genetic information is the claim. And Lenski writes, the authors assert repeatedly, last sentence of their importance statement and first and last paragraphs of their discussion, that, quote, no new genetic information evolved, end quote. However, that statement flatly contradicts the fact that in their experiments and ours, E. coli gained the new ability to grow on citrate in the presence of oxygen. We would further add, which we have not emphasized before, that these CYP+ strains can grow on citrate as a sole carbon source when E. coli grows anaerobically on citrate. It requires a second substrate for growth in order to use the citrate, a phenomenon called co-metabolism. The claim that no new genetic information evolved is based on the fact that the bacteria gained this new ability by rearranging existing structural and regulatory genetic elements. But that's like saying a new book, say Darwin's Origin of Species, when it first appeared in 1859, contains no new information because the text has the same old letters and words that are found in other books. In an evolutionary context, the genome includes not just proteins and patterns of expression, but information about the environments where an organism's ancestors have lived and how to survive and reproduce in those environments by having useful proteins, expressing them under appropriate conditions, but not others, and so on. So when natural selection, that is differential survival and reproduction, favors bacteria whose genomes have mutations that enable them to grow on citrate, these mutations most certainly provide new and useful information to the bacteria. That's how evolution works. It's not as though new genes and functions somehow appear out of thin air. As a bacterial geneticist and Nobel laureate Francois Jacob wrote, Science 1977, quote, "Natural selection does not work as an engineer works. It works like a tinkerer who does not know exactly what he is going to produce, but uses whatever he finds around him, whether he finds whether it be pieces of string, fragments of wood, or old cardboards. In short, it works like a tinkerer who uses everything at his disposal to produce some kind of workable object," end quote. To say there's no new genetic information when a new function has evolved, or even when an existing function has improved, is a red herring that is promulgated by the opponents of evolutionary science. In this regard, it seems relevant to point out that the corresponding author, Scott Minnich, is a fellow of the Discovery Institute and was an expert witness for the losing side that wanted to allow the teaching of intelligent design as an alternative to evolution in public schools in the landmark Kitzmiller v. Dover case. Dr. Minnich, what do you think?
Well, I've read that, and for those of you that are interested, Dusty and I sat down and wrote a response that we included as an epilogue in his dissertation. I could send that out. I mean, the number of comments are legion. This idea of no new genetic information, I mean, we can quibble about what that is, but at some point in evolutionary studies, you have to show a new gene coding for a new protein with new protein folds. Now, the redistribution of information that's already present, in my mind, is not new genetic information. In fact, we qualified this in our paper. Whenever we said new genetic information, we qualified it as a gene with a new function. In this case, you have CYT acting as a citrate antiporter and DCTA as a succinate importer, functioning as their normal protein folding dictates. You've just changed the regulation. Now, E. coli can change regulation of genes easily. I mean, there's nothing new here. Redistribution of genetic information, putting CYT behind a new promoter is nothing new. Now, this idea that, what was the analogy with the book? I mean, I found that analogy just crazy, and saying that it's like saying that Darwin uses the same letters as any other book and therefore doesn't have new information. I mean, that's a straw man argument. We started out with Origin of the Species, the genome of E. coli. We ended up with E. coli's genome with a duplication and a cut and paste error. There's no new idea communicated in this book that and Lenski has generated. I mean, that's a spurious argument. I mentioned before, they brought up, the idea that, well, there is new information because when E. coli grows on citrate anaerobically, it requires a co-metabolite. You have to give it glucose or pyruvate, some other carbon source. That's because it needs the reducing power. As I mentioned in the talk, under anaerobic conditions, citrate's imported, is split to acetate and oxaloacetate, succinate is exported. 4 carbons are lost for every 6 that are brought in. And to keep that transporter functioning, you've got to then reduce oxaloacetate back to succinate. It's a wash. You have to have the co-metabolite because you don't have oxygen present as a final electron acceptor. You're not running respiration. It's simply the fundamental difference between respiration and fermentation. And for them to make this statement really embarrasses me, that it could be so simple and they're missing it.
Yeah.
Okay.
But
do your homework.
Yeah, exactly. And I think the key clincher or refutation of your work, Dr. Minnich, is that you dared to testify at the Kitzbühel Read-Over trial, and that you happen to be a fellow of Discovery Institute. I mean, that's
right. So you impugn my reputation by associating me with my worldview, you know. And I think, again, this is not science. You know, this is embarrassing that people are listening to this. I don't read a paper and then question, was this from a Muslim or, you know, a woman or something else. It has nothing to do. You look at the data. They mentioned in this review article or their blog that our results were unbecoming and unscientific. You know, and why are they unscientific? Because we disagree with them? I mean, I can see where they'd be upset that we repeated their 15 years of work in a matter of days. We got 46 independent mutants and they only got one. And it's the same mechanism involved. And it was inherently obvious. That's why I wrote them in 2012, that there's another explanation, and I got no response. I thought they would do the experiments. I said, it's simply the difference between a direct selection and a screen. You're throwing away these mutants with your daily transfers, and I expected them to do it. And when I went to hear Lenski in Boston, and Hull's work was not even mentioned, I just got mad. And the abstract for this meeting at an international meeting, you know, said this would essentially quench the anti-evolutionists. I'm not an anti-evolutionist. I'm a critic of evolution for the very reason these experiments that he's done have been so overinterpreted. And in our textbooks is the ability of E. coli to evolve new function.
Yeah, absolutely. I agree with you.
So my worldview has nothing to do.
Yeah.
This paper, I specifically requested to be reviewed by John Roth, a member of the National Academy, Tom Silhavi, the editor-in-chief of the Journal of Bacteriology, and then there was one other anonymous reviewer that I don't know. All of them accepted this paper, and our paper was accompanied by a commentary by John Roth agreeing that this whole idea of contingency has to be reinterpreted, that there's another explanation. And he should know. He's the expert on gene duplications and how they occur and their mechanism and how they can generate new phenotypes.
Yeah.
So, you know, Jeffrey Fox said that Lenski's response to our paper was the best post-publication review that he's seen. In my mind, it's embarrassing for the comments that they make, you know. Anyway, I'm mad, as you can see.
Yeah, and rightly so. In fact, Professor Larry Moran of University of Toronto, who's an infamous anti-ID blogger, also promoted the Lenski blog post at his own website, which really shows some of the superficiality of some of the anti-ID crowd when it comes to their ability to critically think.
Yeah, we're called idiots with a capital I and D.
Yeah,
right. Right.
And yeah, it's just remarkable. Anyway, let's take a few questions that have been submitted and then go back to some of Lenski's comments. So an anonymous viewer says, do you think that creationism and religious beliefs about the origins of the universe and life pose a genuine threat to the development and education of science, especially in the US? If so, what should be done about it?
All right, so the question is, does creationism pose a threat to science?
Yeah, do you think that creationism and religious beliefs about the origins of the universe and life pose a genuine threat to the development and education of science, especially in the US?
You know, I'm not an advocate of creationism in the sense of bringing the Bible into the science classroom. It's not necessary. You do the science. You know, the question, and I think that's why ID is attractive to me, it doesn't have any pre-commitment to any religion or set of scripture. It's just asking, can the natural forces of nature, chemistry and physics, produce the complexity that we see on this planet? I think that what we're seeing, and I've told my colleagues that, modern science by necessity, and secular historians agree, even Francois Jacob, who Lenski quoted, in the tinkering comments, agreed in a book written by Harvard University Press that modern science by necessity came out of a Judeo-Christian framework because it believed that the world was created by a rational God in a way that could be understood, and that he was not part of its creation and therefore not subject to its laws. I mean, all of modern science is founded on that profound point. Now, the question is, because it's been so effective, that worldview is so effective in spawning modern science, what happens when we move off the base? And I would say we're seeing it, that I'm the one that's being censored. I'm the one whose integrity is being impugned by my worldview. If we move off that philosophical base, what happens to science? It becomes Darwinian. Whoever has the money and the power are going to dictate what experiments are done and how they're interpreted. And I think we're seeing that now. That science is not about censoring anybody. The whole history of science is one revolution after another. So you go with the data. And I don't think that these long-term evolutionary experiments support the conclusion that they want to see. Right. That E. coli is going to grow in complexity. No, it's stripped down its genome. It's lost information. It's rearranged information that's already present on the chromosome, but there's nothing new.
Yeah. And speaking of religious beliefs, do you happen to know if Rick Lenski is an atheist himself?
If Lenski is an atheist?
Yeah, do you know if he's an atheist?
I have no idea.
Okay, because if he's going to bring our religious beliefs into the picture, then surely his religious beliefs also need to play a role in consideration of his arguments as well, right?
Right.
We had a hand raised from someone. Let me bring up questioner. Hey, you're on with Dr. Minnich. Go ahead.
Oh, Dr. Minnich, I'm Salvador Cordova. I'm actually a friend of Gordon Wilson's. So I just wanted to say hi and thank you for all your work these years. You've been quite an inspiration. I'm now re-entering biology curriculum at the National Institutes of Health. So your discussion is very interesting. I have a couple technical questions. The loss of genes, is that irreversible in E. coli? I noticed that only about 20% of the E. coli genome is conserved. So once an E. coli bacteria loses a gene, in that population, is it expected that the gene loss is permanent?
Yeah, they're two. In isolation, it would be permanent. Okay. The fact that you have lateral gene transfer through conjugation or transduction, some of that can be restored. And we see a lot of lateral gene transfer in E. coli occurring in nature. So, but yeah, if you lose a gene, I mean, it's lost.
And has Dr. Lenski released some of his genomes from the long-term evolution experiment? I can't seem to find it in any sort of genetic bank or anything. Is it only contained in his lab, or is he actually release the entire genomes of the sequences somewhere that we could look at them?
I think you should look into supplementary information in his publications. I think, when you publish, you're required to make this, the sequences available. And we certainly did. I mean, Dusty may know, yeah, I know they're available because we looked at them carefully. So you can email Dusty van Hofwegen and he can get the site for you if you're interested.
Oh, thank you. And one final question regarding, I'm only beginning in my studies in biology. I'm actually a retread from physics. You mentioned there are 46, thank you. Well, I have to thank you for inspiring my interest in these topics. So that's why I decided to go back to school. So thank you very much. Regarding the 46 independent mutants, did the gene duplication occur usually in the same place, or is it more like, now when I looked at the E. coli genome, they're like 50 million bases. Does it actually duplicate in all 50 million spots and then it just happens to be in the right spot? Or does it seem like it optimizes toward one location?
It optimizes towards one location, about 100,000 base pairs spanning the citrate gene cluster, right? And this is being selected for. If you amplify those genes, even though they're expressed anaerobically, any gene is going to be leaky. So the more copies that you have, the leakier the cell is, and those cells can start to grow on citrate, although very slowly. The genome size of E. coli is about 4.6 million nucleotides.
Oh, sorry. Thank you.
Yeah. So, or, 4,200 genes, plus or minus some change. So, but yeah, you see, because you're selecting for these duplications and subsequent amplification in that region. Now, the precursors to our refined mutant that we sequenced, so we could look at the antecedent to our strong citrate mutant. Dusty did this. We just found amplification of that CIT region, but it's a whole population of cells with various copies of CIT-T because they're being amplified. Some may be lost due to the constraints on carrying extra gene copies. So you have kind of a mishmash of population preceding that chromosome remodeling that captured CIT-T behind an oxic promoter.
Well, thank you very much for your time. And it's nice finally meeting you.
My pleasure.
Thank you, Salvador. Okay, let's go through a few more of Lenski's comments that he had to say. So he talks about the claimed rapid evolution of citrate utilization. And he says, in the title of their paper, throughout Van Hofwegen et al. Emphasized that in their experiments, Escherichia coli evolved the ability to grow aerobically on citrate much faster than the 30,000 generations and approximately 15 years that it took in the long-term evolution experiments. That's true, but it also obscures 3 points. First, we already demonstrated in replay experiments that in the right genetic background and by plating on minimal citrate agar, CIT plus mutants sometimes arose in a matter of weeks, Blount et al. 2008. Second, rapid evolution of citrate utilization or any evolution of that function was not a goal of the long-term evolution experiments. So while it is interesting that Van Hofwegen et al. Have identified genetic contexts and ecological conditions that accelerate the emergence of citrate utilization, as did Blount et al. 2008, that in no way undermines the slowness and rarity of the evolution of this function in the context of the long-term evolution experiment, or for that matter, the rarity of CIT plus E. coli in nature and in the lab prior to our work. Third, the fastest time that Van Hofwegen et al. Saw for the CIT plus function to emerge was 19 days from their table 1, and in most cases it took a month or 2. While that's a lot faster than 15 years, it's still much longer than typical direct selections used by microbiologists, where a readily accessible mutation might confer, for example, resistance to an antibiotic after a day or 2. So while we commend the author's patience, we do not think the fact that their experiments produce CIT plus bacteria faster than did the long-term evolution experiment is particularly important, especially since that was not a goal of the long-term evolution experiment, and since we also produced them much faster in replay experiments. However, in a manner that again suggests an ulterior non-scientific motive, they try to undermine the long-term evolution experiment as an exemplar of evolution. The final sentence of their paper reads, quote, "A more accurate, albeit controversial, interpretation of the long-term evolution experiment is that E. coli's capacity to evolve is more limited than currently assumed," end quote. Alas, their conclusion makes no logical sense. If under the right circumstances, the evolution of citrate utilization is more rapid than it is in the long-term evolution experiment, then that means that E. coli's capacity to evolve is more powerful, not more limited than assumed. What do you think, Dr. Minnich?
Okay, well, there's a lot to address there, and like I said, we had addressed all these comments, but there are a few things that I want to say. All right, this idea that it's true, but it's also obscures 3 points. First, we already demonstrated in replay experiments, this is quoting Lenski, in the right genetic background and by plating on minimal citrate agar, CIT+ mutants sometimes arose in a matter of weeks. All right, so I went through those papers and we wrote in their 2008 PNAS paper, Proceedings of the National Academy, they do replay experiments sampling from various generation time points. See their table 1, Blount et al. 2008. And first, they repeated the LTEE protocol using 72 replay populations. They isolated 4 CIT+ variants. These 4 mutants only arose from starting populations taking from LTEE frozen fossils after 30,500 generations. No isolates from 5,000 to 30,000 LTE generation start points. Additionally, these 4 isolates emerged between 750 and 3,700 generations of replay. 750 to 3,700 generations at 6.6 generations per day requires 113 to 565 days respectively for these mutants to appear under standard LTE experimental conditions. Yet they criticize us for taking 19 days from generation 0 without this long exposure to citrate. In their second replay experiment, they took 68 clones from the same LTE generation start points and plated directly onto minimal citrate agar plates and incubated them for long periods. And all after 32,000 generation starting clones. All right, 15 years of constant citrate exposure, and they only got 5. In an expanded version, they isolated an additional 8 CIT+ clones, 2 from their 20,000 generation start point. These are from their frozen fossils. 2 from 27,000 generation start point, and 4 after 31,000 generations. By their calculation, they screened 4 times 10 to the 13th cells in total. So in their combined replay experiments, they acquired 17 additional CIT+ mutants, again compared to our isolation of 46, with a maximum, ours took a maximum of 77 days and only required 12 to 100 generations. In summation, using their same protocol with extended time between transfers, we get the same class of CIT+ mutant in days, not decades, and starting with E. coli, wild type, not a strain that required at a minimum 20,000 generations of constant citrate exposure. They also, what you were reading, by design, the LTE are open-ended experiments to track the evolution of 12 parallel E. coli cultures under controlled conditions. Aerobic citrate utilization was not a specified aim. The highly delayed evolution of CIT+ strains understandably gives the impression that this was a rare innovative evolutionary event. Our studies explain why, highlight why it was not. So we highlighted in our paper that we agree that citrate utilization was not an aim of the LTEE, although it became the most important observation. They're open-ended experiments, and they're the right experiments to do, okay? But we contend that our results do undermine the overall interpretation of the long-term appearance of CIT+ cells in the LTE. Our experiments provide a mechanism explaining why it took so long for this trait to emerge in their experiment, and the explanation is the basis of why John Roth and Meisner-Patton specifically state that the LTE results need to be reinterpreted. I mean, here's somebody from the National Academy agreeing. You got to reinterpret your results. There's one other point that I wanted to make. So they state, third, the fastest time that Van Hofwegen et al. Saw the CIT+ function to emerge was 19 days. And in most cases, it took a month or 2. While that's a lot faster than 15 years, it's still much longer than typical direct selections used by microbiologists where a readily accessible mutation might confer, for example, resistance to antibiotic after a day or 2. I mean, I'm surprised that they make this argument. Resistance to an antibiotic such as streptomycin or rifampin typically requires only a single point mutation. This is genetically equivalent to acquiring bacteriophage resistance, illustrated by Lurie and Delbruck in their seminal fluctuation test back in 1943. This spawned this whole age of molecular genetics. Right? Phage resistance, they showed, requires a mutation that's pre-existing in the population. The frequency of that mutation, for a point mutation, antibiotic resistance or phage resistance, is in the average of 10 to the minus 8, 10 to the minus 9. You can plate a tenth of a mil of E. coli not exposed to antibiotic or a phage and get cultures that are resistant overnight. That's a direct selection. Now, to say that they're surprised it took us 19 days is mixing apples and oranges. You first have to have a gene duplication, all right? And you have to have a duplication of 2 regions of the chromosome, CT and DCTA. All right, that's on the order of 10 to the minus 3, 10 to the minus 4. And then you have to have chromosome remodeling to reduce these duplications to capture a promoter. This is a lot more complicated than a simple direct selection for antibiotic resistance, and shame on them for bringing this up. You know, it doesn't make sense. They're incongruous. They had to screen 10 to the 14th cells to get a single CYT plus mutant. And when you do the calculation, what's required to get a point mutation, a gene duplication, and a promoter capture, it's on the order of 10 to the minus 14. So you could predict the probability of those events occurring in the same cell at the same time in the order of 10 to the minus 14. That's what a screen does. If you allow a progression of events, duplication, more copies, more cells, more targets for subsequent chromosome remodeling, it's going to occur faster, but not in overnight like antibiotic resistance. They're not pre-existing in that population. Only the duplication is. And that's what you start to work with. All right. So again, these are maddening comments that they bring out. They're just not, you know, were they shooting from the hip when they wrote this?
Yeah. Let's take a few more questions that have been submitted. So Rob Ward in New Zealand asks, sorry that I missed the first part of the talk. What was the genome mapped before and after the citrate experiments? Did the mutant bacteria DNA return to their original coding when returned to a non-stressed environment?
So the question is the genetic map. So we had amplification of CT in the production of these mutants and then a promoter capture, and that's going to be stable. I mean, that's fixed. If we haven't done the experiments to look at the fitness cost of that promoter capture, but I don't think it's going to be very large if we grow them continuously in non-selective conditions. I think it would be on the order of 10 to the minus 8, 10 to the minus 9 to lose it. So
cool. An anonymous viewer asks, hi, Dr. Minnich, I may have missed it. Was the population size different between Lenski and your experiments? I assume the effective population size was smaller in the long-term evolution experiment.
No, we're using the same cell densities as they were, the same media with glucose. So it supports about 6.6 generations.
An anonymous viewer asks, what is your opinion on the conference on evolution last November in the UK with large support by ID and third way evolution? Do you think that the landscape of Darwinism is about to shift anytime soon?
I didn't participate, and I haven't followed, just kind of on the surface some of those arguments. You probably know more about that than I do, Jonathan, so maybe your comments are more.
Very briefly, I mean, I was at the conference, and my take-home message from it basically was, okay, so Gerd Muller in the first presentation basically conceded that evolution has an explanatory deficit when it comes to explaining the origins of novelty, which is the key thing we want to explain.
We're dealing with here, right?
Exactly. And then the rest of the conference proceedings was attempting to elucidate or discuss other potential mechanisms that could play a role in evolutionary history. The key elephant in the room, as far as I was concerned across all 3 days, was that yes, those mechanisms that were being put forward were real biological processes which have been observed have been empirically discerned, detected, shown to happen, but there was no empirical or theoretical demonstration that such processes were causally sufficient or adequate to explain what we're trying to explain. They just documented some really interesting processes. There was a very clear absence of an argument presented that these mechanisms actually had the explanatory power necessary. Yeah, in fact, the conference lectures and presentations are uploaded online, so you can go and view those for yourself and judge for yourself. Unfortunately, there was a great deal of hostility, I thought, towards any teleological viewpoint or design viewpoint. There's a couple of episodes where people kind of had an explosion of emotion in the audience about any possibility of design or
Right.
So David asks, I often talk with students at a nearby university about these topics. I often find myself in a conversation that is over my head. Forgive my ignorance, but I'm trying to get a handle on the argument. Could we say that the rapid mutations are the result of existing information present in the genome and not the result of evolution creating new information? Consequently, we would say the information is the result of pre-programming. Thank you.
You know, that's a good question, and it's and this is maybe the point of what you want to extrapolate from the data. So both Lenski's experiments, which I think, again, I don't want to be derogatory on the overall LTEE. I think they're excellent experiments. They're exactly experiments that should be done. And he's to be commended for carrying them out for so long. I mean, it's such a treasure trove of information. But how do you interpret it? You know, both his experiments and my experiments, we start with a CIT minus E. coli, and it adapts through mutation to become CIT plus. So that's evolution, okay? But can you extrapolate that to the question of novelty? Can this answer the long-term requirements of generating new information? And again, new information is a gene that has codes for a new function. And both my experiments and Lenski's with regard to this are lacking. In fact, as Lenski said, the most significant event of these LTEE are the acquisition of citrate utilization. And, you know, I'm sorry, but after 64,000 generations, and you get it once, I'm not impressed. As a microbial geneticist, this is not, you know, a rare, innovative event. It's just reflecting the experimental conditions. E. coli can do it in days versus decades. So that's the whole point. It seems like E. coli can adapt to its environment, but it's got limited avenues by which to do it. And gene amplification, you know, amplifying information that already has in the chromosome, reorganizing that information or distributing that information so that it's now expressed under conditions that it normally wouldn't be is important. Adaptive response, but, you know, I don't think it's anything earth-shaking. Now, if E. coli suddenly starts in their experiments fixing nitrogen, you know, atmospheric nitrogen is the only source of nitrogen required, then, yeah, then we'll pay attention. And, you know, that'll be something, but not this. So there's a limit.
An anonymous viewer asks, a paper was submitted to bioRxiv last year saying that in the long-term evolution experiment, it was core genes that tended to be mutated. Have you seen this, and do you have any thoughts on this? The paper argues that this is due to the atypical environment of the long-term evolution experiment. It seems that the long-term evolution experiment results are not likely to be generalizable and in many respects at all.
Okay, I don't know if I read that specific paper, but I have an idea in terms of the overall results. I mean, Lenski's experiments are beautiful because they're taking isogenic strains of E. coli, 12, you know, starting ancestors that were genetically identical, and running them through a controlled, constant environment and asking what happens. You know, do they evolve? I mean, there's no changing environmental conditions other than the fact that they're being fed every 24 hours. And in that sense, I think it is a beautiful experiment because they do evolve in the sense that, and this is why before the CYT mutation occurred, as I said in the video that we began with, they were in lockstep. They were acquiring mutations in the same set of genes. They were undergoing pretty significant mutations or loss of gene information, deletions, right? They lost motility. They had improvements in bottlenecks for glucose metabolism. So they became specialists in metabolizing glucose. They changed their cell shape. They became larger, increasing the surface area to get more glucose in. So there's change occurring, but ultimately you see improvements that are an advantage to that specific environment, but you're shedding information in the process. You're reducing the size of your chromosome. It's kind of genomic reduction. And in my mind, you know, long term, if you're breeding chihuahuas in a world of wolves, so you know, if you release those back into the environment, they're never going to survive. But they're specialists in terms of the artificial environment that you've made, and they continue to change. They can tweak their TCA cycle to make it more efficient, as they found in these so-called potentiating mutations that Lenski referred to, that he thinks are necessary. I don't think they are. I think they're tweaks that help these organisms, but are not necessary for citrate utilization.
Cool. Let's go back to Rick Lenski and see what else he had to say in response. He talks about the speciation event. He says, to us, one of the most interesting facets of the evolution of the citrate-using E. coli in the long-term evolution experiment is its implication for our understanding of the evolutionary processes by which new species arise. Part of the reason for this interest, and the one that's most easily stated in the popular context, is that the inability to grow on citrate is part of the historical definition for E. coli as a species, going back almost a century. But the deeper interest lies not in labeling a new species or debating where to draw the line between species. Various criteria are used by different scientists, and inevitably, there are many cases that lie in gray areas. Rather, as evolutionary biologists, we are most interested in the process of speciation, the ecological and genetic dynamics that lead to changing biological forms that over time are more and more like a new species until eventually, perhaps far in the future, there is no doubt that a new species has evolved. In short, speciation is not an event. As Patakic and Hankinson, 2009, in Evolution, the First 4 Billion Years, put it, quote, speciation is a series of processes with a beginning stage of initial divergence, a middle stage wherein species-specific characteristics are refined by various forces of evolution, and an endpoint at which a new species becomes a completely separate evolutionary lineage on its own trajectory of evolutionary change with the potential for extinction or further diversification into new lineages, end quote. We realize that scientists, ourselves included, often use shorthand and jargon instead of writing more carefully and precisely. We have no doubt that one can find solid scientific papers that talk about speciation events, but except for cases that involve hybridization leading to polyploids that are reproductively isolated in a single generation, as sometimes occurs in plants, this is simply an imprecise shorthand. In our first paper on the citrate-using E. coli that arose in the long-term evolution experiments, we clearly emphasized that becoming CYT+ was only a first step on the road to possible speciation. One criterion that many biologists would apply to investigate speciation is whether a later form merely replaced an earlier form, evolution without speciation, or alternatively, one lineage split into two lineages that then coexisted, incipient speciation. In fact, we showed that after the new function evolved, the CYT+ and CYT- lineages coexisted, and their coexistence was confirmed using genomic data in Blount et al. 2012. We concluded the 2008 paper by asking explicitly, quote, will the CYT+ and CYT- lineages eventually become distinct species? end quote, and discussing how we might assess their ongoing divergence. By contrast, Van Hofwegen et al. Dismissed the idea of speciation out of hand, not only by calling it an event, but by treating the issue as though it hinges literally on the individual mutations that produce the sit plus cell. For example, they write, quote, because this adaptation did not generate any new genetic information, generation of E. coli sit plus phenotypes in our estimation do not warrant consideration as a speciation event, end quote. And in the penultimate sentence of their paper, they say, quote, we argue that this is not speciation any more than any other regulatory mutant of E. coli, end quote. We also note that this is a rather bizarre generalization, as though the gain of function that gave access to a new resource is equal in regards to its speciation potential to, say, the loss of regulation of a function that is no longer used by a lineage in its current environment. Both might well be adaptations, but one seems much more likely to begin the process of speciation. In conclusion, Van Hofwegen, Hovde, and Minnich have done some interesting experiments that shed further light on the nature of the mutations and ecological conditions that allow E. coli cells to evolve the ability to grow aerobically on citrate, a function that this species cannot ordinarily perform. However, they misunderstand and or misrepresent the relevance of the system for evolutionary biology in several important respects. What do you think?
It's rather long-winded. The bottom line is that we put that in the paper because they speculated in one of their papers that, you know, E. coli has undergone speciation. I read an account, I don't know if it was on a blog, Zachary Blount saying that we may have observed speciation for the first time. That was intimated in one of the quotes that I gave in the introductory, right before our eyes, E. coli's gained this new trait. Richard Lenski wrote an essay that was published in an ASM book talking about his work and saying that this looks like origin of the species to me, you know. So, no, I agree, speciation is very complicated. But outside of the scientific literature, even in Panisi's article in Science, they're hinting. I mean, the fact that, why are they bringing up that E. coli is differentiated from its close relatives by its ability to utilize citrate? And this is a phenotype we've used in the laboratory for over 100 years. Those of you in microbiology remember your IMVIC series in diagnostics, indole, methyl red, Voges, Proskauer, and citrate. You know, E. coli is plus, plus, minus, minus. It's citrate negative. Whereas Enterobacter is citrate plus. It's minus, minus, plus, plus on these four. So it's a phenotype that we differentiated. Now E. coli has changed its phenotype and potentially, you know, how it's going to be diagnosed in a clinical laboratory. So no, I've even seen a textbook in microbiology where they had an insert on Lenski's experiments saying that this is a gain of function. This is, you know, the process of speciation. And I think that's been overinterpreted, maybe outside of the control of Lenski, but we certainly brought it in, although I think he was, and we referenced, you know, the two publications that intimated this was occurring. So I think it's a moot point. I mean, Dr. Bohach and I work with E. coli 0157, A7, the hamburger strain of E. coli. Genome is over 5 million base pairs, you know, compared to 4.2 for wild type E. coli K12. We still class it, you know, as E. coli, even though it's gained over 1 million base pairs of information. So, you know, in microorganisms, how do you delineate one species from another? It's more practical than actual. And even when you have the genomic sequences, it's difficult to do. So, you know, I don't want to, you know, mince words on what speciation is or not. I just see this as a common regulatory mutant of E. coli, and I'm not that impressed by it, nor should I think others be. And there's a simple mechanism that can occur in a short time. The only contingency in these experiments was the fact that they chose to use E. coli B, the Lurian Delbruck strain. We used E. coli K12, although we repeated the experiments with E. coli B. That came out of E. coli K12 came out of Stanford. E. coli B went through, I think, 3 rounds of mutagenesis before distribution to the community for working, doing these types of experiments. And fortunately, it acquired a mutation in the regulator of DCTA. We didn't have to have a point mutation in DCTA, the dicarboxylic acid transporter, to recover succinate. We just had to amplify that gene. They had to have that gene was shut down due to a regulatory mutant that was there to begin with, unbeknownst to them. So there's the contingency. Not that E. coli can't rapidly adapt to citrate utilization.
Yeah, Will McCarroll sent in a question. To follow up on a previous question, I have a layman's understanding that the large majority, if not all, of evolved improvements of organisms occur because of deconstruction of existing information. Yet the controlling scientific community strongly advocates a bottom-up constructive mechanism to produce the life divergence we see today without specifics of how this happens. Are there any examples or studies that demonstrate new information or true creation of new capacity, new capability not related to deconstruction?
Okay, others can chime in on this as well. But I mean, isn't this why these experiments were done? The basic question is, where does new information come from? How is it acquired? Lateral gene transfer, certainly plays an important role. But that's just acquisition of information that's already established. That it's interesting to me that after 12 years of the long-term evolutionary experiments and nothing too exciting was happening, these cultures were all in lockstep, that Lenski teamed up with some computer scientists and Robert Pennock, philosopher of science, and designed, a computer program to demonstrate how information can be generated, this Avita program. And when you don't get it in the laboratory, well, you go to a simulation and they can get it. Of course, people have written about that and where this information is coming from. But no, I don't think there's an example. That's the $64,000 question. Where does novelty come from? You can modify the existing information you have. You can get rid of it. You can amplify it. You can hook it up to new switches in terms of vary when it's expressed. But ultimately, we need a new gene with a new function coming from the existing reservoir of information in the cell.
Yeah. And typically, when we look at cases of antibiotic resistance, as you know better than I do, Dr. Minnich, when you look at the mechanism at the molecular level of how antibiotic resistance is acquired, it's generally speaking modification of a target site rather than generation of new genetic information. Would you say that's accurate, Dr. Minnich?
Yeah, if you look at streptomycin resistance, you have a mutation in a ribosomal protein that renders the cell resistant, but that comes with an extreme fitness cost. You're essentially screwing up the information processing system of protein synthesis, and you get stuttering of the ribosomes, and you double the replication time for these organisms. You have to acquire compensatory mutations, but I think they come with a cost as well. Is it evolution? Is it change over time? You bet. Is it mutation and selection? You bet. It has nothing to do with acquisition of new information or leading to new information. You're just really mucking up what's already there. Ribampin resistance, the targets RNA polymerase, they come with a fitness cost. Fluoroquinolones, gyrase mutations, same thing.
Yeah, which is why information is uniformly and routinely and habitually associated with conscious activity or the role of an intelligent agency. And so it's fascinating that we find points throughout the history of our planet where there are infusions of information to the biosphere such that you have these explosions of new genes and proteins, such as, for instance, at the transition between the prokaryote and the eukaryote, where you see this explosion of new genes and proteins. I mean, how do we explain that in evolutionary terms if we can't, explain how the origins of new genes and proteins? An anonymous viewer asks, back to the population size question, to elaborate, with frequent transfers, i.e. bottlenecks, does this lead to lower effective population size, the harmonic mean, for the long-term evolution experiment compared to your experiment with longer times between transfers? My thought was lower effective population slows down the long-term evolution experiment. Is that reasonable?
Yeah, that's reasonable. I think that's right on. In other words, for these duplications and amplifications in CT, you need longer time in stationary phase. That's the bottleneck. To increase the cell population size, subpopulations of CIT plus cells, and the number of copies of CIT within those cells. So the bottleneck is you're not reaching the threshold. It's like Sisyphus. You're getting close to the top in the LTEE, but as soon as you're close, the stone rolls back down the hill. You recombine out these duplications because now they're a cost. You have to compete with glucose specialists, and you start the game all over. So yeah, it's a bottleneck. By extending the time in the stationary phase, we allow these duplications, amplifications to capture a promoter, fix that phenotype, and you're off to the races.
Okay, let me read the last part of Rick Lenski's blog post. And folks, there's currently no hands raised or questions submitted. So if you want to ask a final question or interact with Dr. Minnich, just click raise hand, or also you can submit question in textual form hitting the Q&A button. Just to finish off the piece from Lenski, so he finishes off with the heading, and the meaning of historical contingency. He writes, the paper by Hofwegen et al. Is accompanied by a commentary by John Roth and Sophie Misnier-Peyton. Their abstract begins, quote, Van Hofwegen et al. Demonstrate that E. coli rapidly evolves ability to use citrate when long selective periods are provided. This contrasts with the extreme delay, 15 years of daily transfers, seen in the long-term evolution experiments of Lenski and co-workers. Their idea of historical contingency may require reinterpretation. Historical contingency is a complicated notion, but it essentially means that history matters. In Blount et al. 2008, we made it clear what we mean by historical contingency in the context of the evolution of the CIT plus lineage in one of the long-term evolution experiment populations. Was this an extremely rare event that could have happened at any time, or did it instead depend on the occurrence of a sequence of events, a particular history, whereby an altered genetic context evolved a potentiated background in which this new function could now evolve? Roth and Misnier-Peyton's suggestion that our idea of historical contingency may require reinterpretation reflects a false dichotomy between historical contingency on the one hand and the effects of different selection schemes on the other. The fact that evolution might be fast and not contingent on genetic background, though the evidence of Van Hofwegen et al. Is at best ambiguous in this regard, in one set of circumstances, has no bearing on whether it is contingent in another set of circumstances. The historical contingency of CIT plus evolution is not mere conjecture. We showed that the evolution of this new function in the long-term evolution experiment was contingent. In replay experiments, Blount et al. 2008 showed that the CIT plus trait arises more often in later generation genetic backgrounds than in the ancestor or early generation backgrounds. Moreover, Blount et al. 2012 performed genetic manipulations and showed that a high copy number plasmid carrying the evolved module that confers a CIT plus function had very different phenotypic effects when put in a CIT minus clone from the lineage within which CIT plus evolved than when placed in the ancestor or even the other late generation lineages not in the line of descent leading to the emergence of the CIT plus bacteria. In the clone on the line of descent, this module conferred strong, immediate, and consistent growth on citrate. In the other genetic backgrounds, growth on citrate was weak, delayed, and or inconsistent. The hypothesis of historical contingency is not mutually exclusive with respect to causal factors of an ecological or genetic nature. It simply says that factors that changed over time were important for the eventual emergence of CIT+. Moreover, historical contingency was invoked and demonstrated in a specific context, namely that of the emergence of CIT+ in the long-term evolution experiments. It does not mean that the emergence of CIT+ is historically contingent in other experimental contexts, nor for that matter that other changes in the long-term evolution experiments are historically contingent. In fact, some other evolved changes in the long-term evolution experiments have been highly predictable and not, or at least not obviously, contingent on prior mutations in the populations. Example, Woods et al., PNAS 2006. For more on historical contingency and the long-term evolution experiments, you can download a preprint of Zach's latest paper from the website. Eric Quandt offers this analogy to illustrate our point that contingency depends on context. Quote, it's kind of like the difference between being an average person attempting to dunk a basketball when all by yourself with unlimited time, and maybe even with a trampoline, versus having to get to the rim in a game with LeBron James and the Cavs playing defense. Just because you can do it by yourself under optimal conditions, does this negate the difficulty of doing it in an NBA game, or say anything about the kind of history, training, and or genetics that you would need for that situation? Dr. Minnich, what do you think?
I don't know. You tell me what you think. I mean, that last analogy, I have no idea what they're saying. LeBron James, and you can do it by yourself. I mean, we did the same experiment. We got the same result 46 times. I mean, what, are we using a trampoline? No,
Yeah.
That's what frustrates me. And historical contingency, well, yeah, who's to say that the 30,000 generations it took is because you had to overcome or suppress deleterious mutations that occurred that blocked the way, that increased the bottleneck. If you start with a wild type E. coli and put it in a medium with citrate, you get there by the same process: gene duplication, amplification, promoter capture, amplification of DCTA. Bottom line, this is what, one NBA team against another. I get frustrated by that. I mean, historical contingency, it implies from their writings that they had to have a series of neutral, maybe even detrimental mutations to get to this final, the last in a long series of mutations to acquire the ability to metabolize CIT. There's no contingency. We could transduce it into the background, from our mutant into a wild type background and get the phenotype. If historically contingent mutants are required for this phenotype, that should not happen, because those mutations are not present in the wild type organism that hasn't gone through these long exposures of citrate. It's a dubious argument.
Yeah.
It's frustrating.
Yeah, I agree. Royal Truman asks, the controversy seems to be a lot less about science than the storyline or significance placed on these experiments. So he says, I guess you would agree with that?
I do agree with it. It's how you interpret the process. I mean, if you're pre-committed to a materialistic evolutionary viewpoint, and you're doing these experiments, and you see a new gain-of-function mutation, a rare new gain-of-function mutation, E. coli now can use citrate, and we define it as citrate negative, then you're going to be excited, because this is what you've got to see at some point. You want to see it. It's a rare contingent event that took the equivalent of, a million years of human evolution. And you can't, it only occurred once out of 12 identical populations. It gives that impression. But I think when and that's what prompted us to do the experiments, that we thought, well, if gene duplications are occurring, these are inherently unstable in their early stage of production. we should be able to get to the same point easily. We should get, which we did, dealt a royal flush every time. You know, this isn't luck and skill. This is just microbial genetics.
Yeah.
Period.
We have two hands raised. We'll take those. Bill Cole, and then we'll have Royal Truman. Go ahead, Bill.
Yeah, Scott, I just wanted to say, because I can see your frustration and feel your frustration. When I first looked at this, I was looking for the evolution of a enzyme, right? Whether they found an enzyme. When they didn't find an enzyme, it was pretty much discounted in my mind as being a very significant evolutionary event. Anyway, what I wanted to ask is more on the social side, and just to maybe think through some of your frustration here, which is when we talk about evolution, and we're apologists or whatever, we're talking about we're talking about two issues. One is a worldview issue, and the other one is a funding issue. Because if we come up with data that says that evolution is difficult, we're attacking this thing on two fronts that are very hard, right? And so there is a very difference from you getting a critique from Richard Lenski on an evolutionary project where you discount his project versus many of the papers you've done in your life where there's no real threat. You guys are just talking about something going on or trying to figure out what's going on with the bacteria itself, right?
Right.
And so maybe you could think about articulate, the different view, because I know you've been on one side with Lenski and with also the bacterial flagellum, where you're attacking worldview and funding, right? Versus just what life is like normally when you're just having scientific discussions over. You may have disagreements, but they're not worldview or funding disagreements. Fundamental.
Right. And I think that's a good point. And I think, I hope I'm not coming across as too dogmatic, but I am frustrated. I remember reading Lenski's first paper where they acquired CIT+. They hadn't described the mutation, the pathway. And in the introduction, they said, this is so rare, it's only occurred once before. And they cite Barry Hall's paper. They don't give any details. I read Barry Hall's paper that was published in 1982, and suddenly, you find that E. coli can grow on citrate anaerobically. You read the papers by Lunkin and Galtshaw. I went and bet a friend at my university that's part of Beacon, part of Lenski's, NSF-funded Beacon project, James Foster, and I said, has Lenski figured out the mutation that accounts for CIT+? He said, no, I don't think so. And I said, I'll bet you a beer. This is 2009 or 2010. I'll bet you a beer. When they figured out it's going to be, they captured CIT aerobically. They got the anaerobic transporter. It was that easy, Why, from my perspective as a microbial geneticist and an ID person, you just cut to the chase and say, it has the capability, that's what they're going to find. And they find it, and they still persist in saying that this is a novel gain-of-function acquisition. And I don't see it like that. So it comes down to worldviews. And I would say that their experiments tell me, over the long run, E. coli can adapt through mutation to this environment, continue to adapt, although it's slowed down. They want to say it's going to continue, but what's happened? The bottom line is you got genomic reduction in a couple of regulatory mutants. There's no new genetic information, period. And you've got to account for the diversity on this planet at some point on how you're getting these new genes that have totally new functions from the reservoir of information available to you.
Yeah,
by and large, this experiment is leading E. coli down a genetic box canyon. It's not into new heights and new planes where it's going to diversify.
Well, their only out is to define information in a very tight window. And as you said before, that's quibbling, right? It's
Right.
quibbling. It's not. It's trying to discount your no new versus very little, or however you define that. They're trying to take your argument and box it into some little solution, but in reality, it's not relevant to the discussion.
But I think that's the point that it is. You can distribute the information that you have, maybe express it out of context, get a cut and paste error, but it's not something you still have CIT transporting citrate and exporting succinate, and you still have DCTA importing succinate to recover those 4 carbons that are lost. Right. Bottom line.
But no new enzyme.
Right. Exactly.
Okay.
Thanks for your comments.
Yeah.
Appreciate it.
Thank you, Phil. Let's lastly go to Royal Truman. Royal, are you there?
Yes, I'm still here. Can you hear me?
Yeah, we can. Go ahead.
Okay, Scott, thank you very much for the presentation. I had been following the science part with a lot of interest, but I think what you've done is so important to put things in real and proper perspective, which is what a lot of people can't do. They don't recognize the points you brought up about genomic reduction being really what you see in these experiments, or that all the functions are already there, but they have been specialized or emphasized, but they're not new. Really, really important things. I think the only thing I can comment to add to that is maybe when we write papers or books, it's very common for us to take an existing sentence and change the order. It changes nuance a little bit. We might even replicate a sentence to add emphasis, but I would agree with you that would be nothing truly new, nothing significant. That a series of letters, which was Lenski's example, that a book is a book in your argument, and that all you're doing is throwing a bunch of letters is silly, because what is happening is the informational content of sentences or phrases are being rearranged. What is missing are entire new thoughts, if you want to use a book analogy. And that's what the Lenski's would have to come up with. A brand new function that wasn't involved in any existing metabolic processes at all. That would be something totally new, but not, a barely measurable function that's rarely seen, has been emphasized in an artificial situation at the expense of a lot of other functions like mobility of things.
Right.
So you almost have to play a lawyer. Your job is almost to play a lawyer to argue effectively of significance. It's not even the science of things that's argued.
Yeah, I think that's well, I want to, going back to your comment, I have in Dusty's epilogue. So Lenski wrote, the claim that no new genetic information evolved is based on the fact that the bacteria gained this new ability by rearranging existing structural and regulatory genetic elements. But that's like saying a new book, say Darwin's Origin of the Species, when it first appeared in 1859, contains no new information because the text has the same old letters and words that were found in other books. And so we would respond, this analogy is nothing but a straw man argument. A more correct interpretation using this analogy would be the following. We started with Darwin's Origin of the Species, i.e. the E. coli chromosome, and after printing millions of copies, we found production errors where several pages had been duplicated, and some of these duplications had been edited out, leaving a cut and paste error at the duplication junction. We started with the origin of the species and ended up with the origin of the species with no new information. In other words, we didn't end up with a copy where Darwin's Origin of the Species presents a new argument. It's simply the same book with a deletion, cut and paste error, and a duplication. In short, no new information. So I don't know where he's coming from that analogy. This like, because it has the same words in other books. No, that's totally missing the point. We want a new idea.
That's correct. I think it's important to always remind everybody that bacteria are extremely adaptable. It's not evolution. It's part of the range of behavior that's foreseen. Huge populations that already have many, many point mutations, they're going to adjust to circumstances. And that's not, it's not really the information argument at all.
In a sense, although you've got a limited reservoir of information, but in terms of evolution, yeah, when you throw in lateral gene transfer, E. coli will mate with anything. And so you can get a lot of exchange of pre-existing information. But the bottom line of the novel information, de novo, is still, what's required.
It reminds me a lot of computer science, You've got like exo functions, which are already there. You can combine them in interesting ways. You can appear to get something that's, novel, but at some point, those new functions have to be programmed. You have a function that does a factorial calculation, you've got a function that does a combinatorial, whatever, and you sort of call the functions and you use them, but they're already there. You're just rearranging what's there.
Right. There was a programmer behind it.
Yeah.
That's the point.
Just before we close, let me just take one more hand from Charisse, who's a biochemist. Charisse, are you with us?
Yeah, can you hear me?
Yeah, we can. Go ahead.
Hi, Dr. Minnich, how are you?
I'm doing well, thanks.
I actually just wanted to see what you thought of the idea that we should probably be more specific when we talk about, the idea of novelty and information and things like that is not so simple. We had the pleasure of hearing from, oh my gosh, what's his name? Jonathan Wells, regarding information that's not even a part of DNA at all, regarding development. And so I would just taking the devil's advocate position of, okay, if there's already, we have different types of information already within just like the genome itself, we have like sequence. And I feel like what people, when I heard that result, it's like, okay, you evolved something new, novel. I'm also thinking of, oh, you have like a new invention, you have a brand new, like functional unit that wasn't there before in any way, shape, or form. Like, for example, a new enzyme. Like, I would be very impressed if this thing invented a new enzyme out of completely brand new sequence space from anything that's already in the genome, right? And that's an important problem that needs solving, because that is what it looks like has happened through evolutionary history, and is that novel sequence space with novel function is present in taxonomically restricted. Anyway, so, but what I feel like somebody could say is that, okay, well, in the, like, if you were to compare across species, what you not only find is different sequences, you also find different organization strategies, like, of genetic information and ways of controlling and regulating and things like that. Which, wouldn't it be fair to say that there's information, that's another type of information, right? Is
right.
structural and stuff like that. And that this whole idea of like genome editing being like something that it seems like cells are doing, I guess, is it worth it to try and tease apart, like, what, more clearly, like, okay, this is a win for evolution in the sense that apparently you can do this type of, you can have this type of information be added into the system. And yet we still have yet to see in real time novel sequence space that makes a new enzyme be added into the system.
Right, and that's the central question. I agree. I think we can get bogged down into definitions of information and Shannon information, whatever. And I'm not an expert in that area, but I tried to qualify it in this paper as new gene with a novel function. Now, I think that, we may find, John Roth had a paper a few years ago on enzymes required for histidine biosynthesis, one of his areas of expertise. And one enzyme appears to have, two substrate specificities. One is extremely weak, one is strong. But if you allow that gene to duplicate, now you have specialization that can occur. And in that case, it could be, evolution of new information. But the fact that it had residual activity, I think you can easily perfect that with all the protein folds that are required. But he cites that work in his commentary to our paper, and I think it's something we should carefully think about, because gene duplication, when you have multiple copies, you can fix one that's required, function that's required, and the other one has now some liberty to experiment and change. And can it acquire a new function de novo or whatever? I think that's a significant area that we will pay attention to, and it could be a win. So anyway, but I appreciate your question. I think we have to be very careful and precise in our definitions, and information is one of those areas that can be kind of plastic.
Right. And I think it's worthwhile too, because of the edge of evolution type of question, right? Like, we're not saying that, Noah, and I know you're not saying that, but how do we help draw the distinction? Because I feel like it is really hard to explain what we mean when we say biological information, and because we are talking about something immaterial, and that in and of itself becomes difficult to explain. And then if it seems like we've rigidly defined it in one particular way, I feel like that becomes harder. And yeah, it's essential to then think about maybe designed plasticity within, you know what I mean?
Right.
How can we detect that?
You can look at it at the genome. It's a reservoir of digital information. Nucleotide sequence is digital information. But when you start looking at the three-dimensional structure of DNA, then you have analog information, where the structures is essential too, and that can change. And then you throw epigenetics and DNA methylation on top of that. There's another layer to the onion that is involved, and it is a complex problem. Right.
And I think it would be worth it to see if there's signatures of design, even in the ability of to what would it look like for this maybe to have been facilitated, this type of an adaptation that they're finding. What would it look like for this to have, for the capacity to do this, to be directed even, what kinds of signals would you be looking for, kind of thing. And right. Yeah, so.
And genome organization has to fall into it too.
Right.
It's not, we now know that the position on the chromosome is not arbitrary for a given gene.
Exactly.
Why is it, if all these genes are evolving, with no direction, contingency occurring anywhere on the chromosome, what brings them all into juxtaposition next to each other in the E. coli chromosome, where you have all enzymatic functions for histidine biosynthesis or arabinose utilization or beta-galactosidase utilization? Why are they all clustered together? What selected force is aligning all these things in an optimum position for their expression, when, if they're happening through the evolution of some other information on the chromosome, they're locked there? What you have to say, well, recombination is occurring, but what we're seeing is more in time, it looks like these chromosomes are, flying apart in terms of inversions and everything. So there's another layer of information. Is not only what genes are present, but where they're positioned on the chromosome.
One can
Beautiful studies going on in terms of, catabolic genes are organized near the terminus, anabolic genes near the origin of replication, and that's because they all are keying off the information of DNA supercoiling, which is dependent upon ATP levels. So you have this feedback going on, and I think it'll be interesting to do experiments where you take a key gene and put it somewhere else in the chromosome, and how does it affect its regulation?
One can extend that argument to flagellar assembly, of course, which you know quite a bit about as well, where you actually have the genes necessary for synthesizing different parts of the flagellum clustered together. So you have in the sort of paradigm system in E. coli and Salmonella, you have the three classes of genes: the class 1, class 2, and class 3. Although the class 1 genes is only 2 genes over 1 operon, you have the class 2 genes are 35 different genes over 8 operons. And what leads to the clustering of those genes, right? Because if you're simply just grabbing proteins from different systems and putting them together to make a new novel system, then what is also facilitating the particular clustering on the operons that makes their regulated expression possible.
Right, and even genes within an operon. So since RNA is degraded from the 3' end, you find components of the flagellum that are required in lesser numbers, for ring protein or hook or whatever. Those genes tend to be on the end of the operon versus, near the 5' end. So yeah, all kinds of interesting.
And your friend Dusty just raised his hand if you want to say hi to old Dusty.
Sure.
Hi, Dusty. Are you there, Dusty? Okay.
Okay. Well, hi to Dusty. He did a good job on this work, and of course, he should get back in the lab over there in Montana. And
okay. Are you there, Dusty? Okay, maybe not. Okay. Anyway, it's been really great to have you on, Dr. Minnich. Thanks for joining us.
Okay, and thanks everybody for participating in good questions. And if you have any comments, email me. Be happy to carry the conversation on.
Yep. Do you want me to give your email address in the chat window, or
yeah, you could do that.
Okay.
It's just sminnich@uidaho.edu.
Okay.
Okay.
Cool.
All right. So can we sign off?
Yes. Okay. Thanks so much for joining us, and it's been a real pleasure.
Well, thanks for your work and hosting it.
You're welcome.
Good job.
Yeah, we must have you on again sometime in the future.
Okay.
All right.
Yeah.
Bye.
Later.
Bye.